So today we're just going to be introducing microscopes. And so microscopes have not been around forever. They were kind of invented in 16 and 1700s. And you know, what development, what technology did people have to really figure out before they could create the first microscopes? It didn't look like this. They didn't even look like this old one. They looked a lot different. But what's the key element to a microscope? Olivia? Exactly. What do we call that glass part?
The eye.
Well, there's an eyepiece, but just a general. Not quite. There's another term I'm looking for, Caleb. A lens. So it was really, it took people to figure out how to make a good lens. A lens is basically a piece of glass, or nowadays it could be plastic, that's formed into a certain shape that gives it certain properties that allows it to bend light in a certain way. Some lenses can magnify an image, and it wasn't until people really figured out how to create good lenses that they could create the very first microscopes. So the very first microscopes, some looked like this, okay? That's a little bit later on. Some looked much different, but they all use lenses to magnify an image. In fact, scientists couldn't really explore or identify cells until the microscope was invented and kind of was made good enough to see cells. Because, like you guys told me in the beginning of class today, microscopes are used to see things that are often too small to see with your regular eyes. And so people didn't know about many of the types of small living creatures in pond water. They didn't know that blood is made up of small little red discs that we call blood cells. So it wasn't until scientists had these microscopes they could figure some of these things out. So microscopes are important. Here in these diagrams you have an older sort of antique microscope and a more modern microscope. Here you have a couple of images as they are viewed through the microscope. You have some plant cells here with green chloroplast, and you also have a letter E that is shown here upside down and flipped and inverted because that's what the microscope does. Because of the way the lenses are arranged inside of the microscope, images get flipped left to right and also inverted upside down. All right, so let's talk about, so this unit, we're going to start by reviewing microscopes and what microscopes do. We're going to practice using microscopes before we actually start moving into looking at cells and talking about cells. We need to learn how to use the microscope first. So we have some vocabulary. This is all related to cells. We're not going to go over it now. It's there for your reference as we move on in our unit. Okay, so let's just introduce three types of microscopes. We'll be using two of the three. The first type is the kind we will be using most often. It is called a compound light microscope. This is a compound light microscope I have up here. The compound microscope uses obviously light to create an image, and it's called compound because it has multiple lenses that magnify the image. The microscopes we use can give us a magnification of between 40 and 400 times. So that means when you look at an image in the microscope, what you see is 40 times larger than the actual size, up to 400 times larger than actual size. So that's how much magnification our microscopes give you. Some light microscopes can give you a greater magnification, up to maybe 1,000 or so, depending on the type of microscope.
Is there any more? Like
Yeah, there's different ways, different types of microscopes that can give a higher magnification, but they're not as common. So that's the compound light microscope. Another type of microscope is called the dissecting microscope, even though it's used for many things, not just dissecting, it's used for many purposes. But it's called the dissecting microscope. It works differently than the compound light microscope, and we'll talk about that. Here is an example of the compound microscope. The dissecting microscope. You'll notice it has two eyepieces. The base looks different. Okay. One other thing you'll notice is that in the dissecting microscope, the light comes from a different place. So on our compound microscope, okay, so our compound microscope here, you can see there's a light source down here. This light shines through the stage, through a little piece of glass. You put your slide here. The light will go through the object you're looking at on your slide. Then it goes through the objective lens, up through the eyepiece, and to your eye.
Are we gonna be
That's the path light travels. Now, if you look at this dissecting microscope, it works differently. The light comes from up top here. I don't know if you can see that it's lit here.
Yeah.
So the light comes from here, bounces off what you're looking at, and then travels through these lenses here and the eyepieces here. So the light comes from two areas. That's an important difference because on this microscope, for example, the compound microscope, could I look at my hand through this?
No.
Yes. I could not. Why could I not? Yeah?
The light went up here.
Yes, my hand is too thick. It blocks the light from traveling through and up through the eyepiece. I wouldn't see anything. So you can't see a thick object, an opaque, we would say, object with this compound microscope. Anything you look at has to be very, very thin. Thin enough that light can pass through it. So it only is used to see very thin objects. However, the dissecting microscope, because the light bounces off the object, again, this doesn't have the right adapter, so the light's not as bright as it should be. But if I put my hand here, again, I can see my hand with this dissecting microscope. Light will bounce off of my hand and then up through the lenses. So I would be able to see a three-dimensional object using this dissecting microscope. So that's a big difference. The dissecting microscope doesn't give as great a magnification. It goes from about 10 to maybe 40 times.
Mr. Dura?
Yes.
Are we ever going to be a dissecting anything?
Yeah, later in the year, in the spring.
Frog?
Yes. So we have a compound light microscope. Don't worry about it now. A dissecting microscope. And then the third type we actually will not use,
Scientific.
but we're going to learn about. This is what it looks like. It actually looks more like a computer than it does a microscope. It's called an electron microscope. And instead of using light to view an image, to create an image, it uses a beam of tiny little particles called electrons. And it can make a very fine image. It can magnify to a much greater degree. It can magnify up to 1,000,000 times.
Whoa.
So an electron microscope is a larger microscope. It's much more expensive. You need special tools to use it, but it can give a high level of magnification. There are other types of microscopes. We won't talk about all of them, but I just want you to know about these three. So if we look at some images that are taken from these microscopes, when you look at an object in a compound microscope, it gives you a very flat view, a two-dimensional view of the object. First of all, because the objects you're looking at have to be sliced very thin, almost paper thin, that there's not a lot of depth to them. This is an amoeba. It's a living organism, a single-celled living thing. We will look at living amoeba later in the year.
Ew. Where do they live?
This is another single-celled protist. This is a paramecium. Again, it's a single cell, but it's alive. It's moving. You can see it move through the water. These are the kind of things scientists didn't know existed until microscopes were invented, because these are very, very small organisms that you cannot see really with your naked eye. So once they started looking at things in the microscope, they started to discover that there is life that's much smaller than we had thought about.
Wait, do we like touch them with daily baby?
No, you don't. Oh, well, yeah, you go swimming out of a lake or something. There are some of these protists living in there. They're around you.
It's just
These are Vorticella, Radiolarans, Foraminiferans. They're all just various types of very, very small living organisms that we can see using the compound microscope. But notice, all these views, things look pretty flat, pretty two-dimensional. Versus the dissecting microscope, which allows us to see a much more three-dimensional view. It gives us much better what we call depth perception. Here we see an aphid,
we're learning about that art.
a small little insect crawling across a ruler, and you could see there's shadows. You could see it's three-dimensional, and again, that's because the light is coming from the top and bouncing off of it, and also because of the two eyepieces, which we'll talk about in a second. This is a planaria. It's a flatworm. We will look at that planaria later in the year. This is a seed just beginning to grow, a seed that is germinating and breaking out of its seed coat. So one question is, why does the dissecting microscope give us a much greater sense of depth versus the compound microscope? And the reason is because of the two eyepieces. Just take a little digression here. We see humans see pretty well three-dimensionally. We have good depth perception. Right? Some types of animals do not have good depth perception. If you take, for example, a goldfish. What's the difference? You know, why would a goldfish not have good depth perception, but a human would? Right?
Because their eyes are on the side of their head, and while we're like this, we see straight, and they see probably side to side.
That's exactly correct. So having eyes on the side of their head allows sort of a goldfish. They have a very good, you know what we call this?
Air.
Vision on the sides.
Oh,
Yes. Peripheral vision. Because their eyes are on the side, they can see a huge, wide area. Us, on the other hand, both of our eyes face in the same direction, so we can't see as great a depth perception. We can see not probably quite even 180 degrees. But having two eyes facing the same way is what gives us good depth perception. So do a little test here with me. Close one eye, close your left eye. Put your finger close to your right eye, in front of it. And then switch eyes. Close, open and close your eyes. You've probably done this before, right?
Oh, yeah.
Has everyone done this, or is it just me? Okay, so what seems to happen? So when you look at it with one eye, it's sort of right in front of you. When you switch to your left eye, where is it?
Side.
Off to the side. Why? Your finger's not actually moving.
Your eyes are. No,
Ryan?
angles.
You can't see it from how close it is in that one eye. It's kind of hard to see it.
Okay. And somebody else said, Neve, what did you say?
The which one?
The last thing you said.
It switches angles?
It switches angles. So when you have this finger in front of your right eye, close to it, your right eye is looking at it, sees it straight on in front of us. But your left eye is looking at it at an angle, so it appears to be off to the right. And so when you switch your eyes, you see it moving. Now, when you hold it out like an arm's length, do the same thing. It still moves, but how much does it move?
Not a lot.
Not as much. Because now your left eye has a different angle to see. It still looks like it's in slightly different places, but not as much. If you look all the way across the courtyard at one of those up and down vertical beams and do it, they don't really seem to move at all when you switch right to left. So your brain is able to use this information. Having two eyes facing the same direction but next to each other, each eye sees objects at a slightly different angle. And our brain knows this. Our brain tells us if my left eye and right eye see this and it looks like it's in very different places, this object must be very close to me. If I look at something and I see that it doesn't move at all, both my eyes basically see it in the same place, my brain says that object must be far away. So our brains interpret this information and allow us to get a good sense of how near or how far something is. This is also how 3D movies work. When you go to a 3D movie, you have to wear those glasses because they allow, because of the polarization of the filters, they allow each of your eyes to actually see a different image. And so when the filmmaker wants an object to appear like it's close to you and popping out of the screen, they make it so each eye sees the object at a very different angle. If they want something to appear far away, they allow both eyes to basically see it in the same place. Or if you ever looked at those red and blue drawings and you put the 3D glasses on, if you take them off and you notice, the picture is drawn twice, once in red and once in blue. And the areas when the red and blue are very close together, those are the things that look far away. If the red and blue lines are far apart, those are the things that appear to be close to you in that 3D image. So notice that. Next time you see one of those 3D drawings, check it out and you'll know now why some things appear to be close and some things appear to be far. So my whole digression is to tell you this has two eyepieces, allowing both our eyes to see the image at a different angle, giving us good three-dimensional view. The electron microscope, that gives us great magnification. We can see things close up. Usually the images are gray. You can't really look at living cells or living organisms with an electron microscope because the objects need to be coated in metal so that the electrons bounce off to form the image. So usually the things are not alive and they have to be coated in metal. But here's what a spider would look like.
Ew.
That's toilet paper. That's Velcro. That's a human hair.
Ew.
Funny.
Split end.
That's the foot of a fly. All these tiny little hairs helping it to stick to a surface. This is a fly. You can see its eyes, but on top of the fly's head are tiny little parasitic mites, tiny little insects that are colored in green here. Here's another mite that's on a beetle.
That is ew.
Mites are just tiny little insects that live on other insects in this case. The colors are artificial. So the electron microscope gives a black and white image. Sometimes people color them various colors to make things stand out more. Here's a whole bunch of insects under the electron microscope. It's really cool to look at insects and see all the different mouth parts and their eyes and stuff.
Those are real?
Yeah, these are all real.
I don't want them in real
What?
life.
Think of
Oh, he's here.
a mantis.
Oh, that's cool.
Yep, fly, yes. A bee, spider,
Oh.
ant, grasshopper,
They're disgusting.
fly.
Is it dead?
Yes. They're all dead, but that one looks dead. All right. So what we need to do now is, and this is what most of your quiz on Monday is going to be, about the parts of a microscope, where they are located, and what they do. So that's what we're going to finish up today with, talking about this microscope. Now you have a drawing here. This is the same type of drawing that will be on your quiz. But this microscope has a large number of differences with the microscopes that we're actually going to use. So I'll point some of those out to you right now. All right, so if this is our microscope, if we start, when we look at an object that's on the stage, the object gets magnified by two different lenses. What's the first lens? As light's coming from our light source through here, what's the first lens that will magnify the image? Where is it located on here? Anthony?
The one like in the middle.
Yeah, this one. So as light travels, it'll go through one of these lenses, and that magnifies the image. The light continues up. It bounces off a mirror here. Then it continues up, and it goes through another lens at the top, which magnifies the image again. Okay? So those are the two lenses that magnify the image. The one you look through is called the eyepiece, and these ones down here, that you can select different ones, that's called the objective lens. So the eyepiece, and we have the objective lens. Now this microscope in your drawing, it's straight up and down. Our microscopes come back at an angle, but it's the same thing. Ours use a mirror in here to redirect the light. These, the light just travels straight through. So each of those magnifies our image. And you'll notice on this microscope there are 3 objective lenses. There's a medium power objective, a low power, and a high power. And you can select different lenses depending on how much magnification you want. You can select different lenses by rotating this round piece here. And that rotates the objectives. Now listen, you could hear as you get a lens in place, it clicks. One thing to be careful of as you're switching from one power to another is sometimes people don't actually click it in, and then the objective lens is not in the correct alignment and you won't see anything. You have to make sure the objectives are clicked in as you're looking at your specimens. The part you rotate has a name. It's called the nose piece. Jake? No, it'll just be random.
Would it be like a word name for it?
It'll probably be matching. So you'll have to match the name of the part to where it's located. The tube that separates the eyepiece from the objective lens is called the body tube. It makes sure that there's enough distance for the light to travel for it to create a clear image. Now the microscope in the drawing down here, it looks different from our microscopes. What does this look like here? That's a mirror. Older microscopes often had a mirror down here. And what you would do is you'd have a separate light that you plug in. You aim the light at the mirror. The light bounces off and then goes through this microscope. Newer microscopes don't have a mirror there. They just have a light that shines directly up through the stage. So that's the light source that's here. In this one, it's a mirror. In ours, it's an actual LED light. Then you have this flat part. So when we use the compound microscope, we look at what we call a slide, a piece of glass where we put our specimens, and we place that slide on this flat part. It's called the stage. You guys use microscopes in elementary school?
Yeah.
No.
Some did, but you're in a different district, right?
Yeah.
So the part you put your microscope on is called the stage. And some microscopes have 2 metal clips here that hold the slide in place so it doesn't move around too much. They're called the stage clips. Our microscope's different though. So like on this microscope, this has these stage clips. Okay, to hold something down. But these microscopes are set up differently. If you'll notice, this compound microscope has this a clip here. This is the stage clip. But then it has all this other stuff attached. This is something called the mechanical stage. What you do is you clip your slide in, and then to move the slide around, you use 2 knobs. This knob, if you look closely, what does it do? It moves the stage, mechanical stage forward and backward. This one moves it left and right. So you don't have to move it around with your fingers like you would with that microscope. You actually use these knobs to move it, and allows you to move it much more slowly in a much more controlled way. Because sometimes when you're looking at something, if you have to move it with your fingers, you like move it too much, and it comes out of view. So this is a nice system that we have on these microscopes, but it's not shown here. Underneath the stage, there's this dial. Paige, what's on the bottom of that dial? Different sized holes. So as I select different sized holes, what do you think that changes? This is on the bottom of the stage, and the holes are different sizes. Matt?
How the light reflects?
Yeah, a small hole allows less light through, which makes the image dimmer, versus if you select the larger opening, it allows more light through and the image is brighter. Sometimes you want a brighter image, sometimes you don't. Sometimes it's too bright, it hurts your eyes. Sometimes it's too dark and you need to see things better. So you can adjust the amount of light using that dial. And the name of that dial, it's called the diaphragm. Then there's a couple parts that are basically for support. You know, there's the part that you kind of hold on to here. It supports this part of the microscope. It's here. It's kind of like a handle that you might use. That's called the arm of the microscope. And then the part that rests on the table is called the base of the microscope. And then we have the two last parts. So these two knobs, on this microscope, they're two separate knobs. They're very important. Those knobs allow you to focus your microscope. So when we say focus the microscope, it means to make the image clear.
That's big.
Yeah, and the image is only clear when things are aligned properly. So, for example, on this microscope, if I move this knob, what happens?
It raises it.
Yeah, the stage moves up and down. And once the stage gets to the proper level, what you're looking at would be in focus. Okay? So those knobs are used for. Now, if I move this, you can see it moves. What about when I move this other one? Does it seem like anything's happening?
Yeah, doesn't it do something with
It's doing actually the same thing. It's moving the stage. But it moves at a much, much smaller amount. This one, the larger knob, that moves it a lot, is called the coarse adjustment. It moves things a lot. It gets the object in focus quickly because it moves the stage a lot. The fine adjustment also moves the stage, but only a very, very small amount. So when you're using the microscope, usually you get it sort of in focus as best you can with the coarse adjustment, and then you can kind of fine-tune it by using the fine adjustment knob. The other thing that's important, when you have it on the highest power objective, the longest one, it's pretty close to the stage. And if you use the coarse adjustment, you could actually hit the stage into the objective and break your slide. So when we switch to high power, we only use the fine adjustment to move at small amounts. Because if we use the coarse adjustment and crank on it, we can break things. Okay?
I broke a slide last year one time.
So that's the way this image is set up. We have a coarse adjustment, a large knob, a fine adjustment, the smaller knob. But when you look at the microscopes we're going to be using, like this one, there's only one knob, right? There's this one. So watch as I move this knob. You can see the stage goes up and goes down. So where's the fine adjustment on this? Here?
Yeah.
Nope.
Okay.
That's for my stage.
Wait, and right there.
Here?
No.
It's right here. So this knob is two parts, the larger outer portion, and then the smaller knob, which also moves the stage, but it moves it smaller amount at a time. So this one has the coarse adjustment and the fine adjustment built into one knob. It's called a coaxial filter.
Where can you buy a microscope?
Usually there's like catalogs of science supply stores that sell them. You can buy like up here. Somebody brought this in one time. It was like a toy microscope they had when they were younger. You know, and these you could buy any like Toys R Us or something. The problem is that they don't work very well. They're made of plastic, and you could get some of an image, but oftentimes the lenses, they're made of plastic. They break very easily. And they don't give you a very good image, but you can sometimes see things through these microscopes. So this one says it goes up to 900x. Okay, almost double what our microscopes do.
Probably not clear.
Yeah, it's not clear at all. It has very poor resolving power. All right, so tomorrow we'll actually get to use some of these microscopes and look at some things under them. But those are the parts, and that's going to be the focus of your quiz on Monday.